that the recording just cut off right at the end, but I was looking, it was like, I think it was like 4 slides, 5 slides maybe that the recording missed, and it was how viruses are grown, which is just that one PowerPoint slide, really easy. Then the graph thing that we went over, understanding acute, latent, and then oncogenic viruses, which again is just like one question, kind of generally understanding what's involved in that. I'm sure the book gets in a lot more detail, but the main thing with oncogenic viruses is that they're usually the viruses that put their genome into our genome, and they'll activate oncogenes that are already in our genome. And I think I forgot to mention yesterday, but in rare cases, some viruses contain oncogenes themselves as well in their genome, that when they get in ours, then they lead to the transformation of our cells in cancer. But otherwise, we have these oncogenes as well as just in our own DNA, and it's only if they get activated that it can lead to cancer. Yeah, that led all kinds of questions. I think Jessica did it first.
I was reading also in your PowerPoint that the oncogenes, they display abnormal proteins, and then that's what causes the tumor.
Right. So just that transformation of cells, they're abnormal. They're displaying abnormal proteins. They're growing at abnormal rates and abnormal patterns and fashions, and all that's what leads to a tumor. Yeah. Number 10, was there another question on oncogenes? I saw a couple other hands go up while we were talking about that. All right, good, it was covered. So number 10, how does replication of bacteriophages differ from animal virus replication? So there was one step missing in bacteriophage. Uncoding, right? And the difference is because of the entry. Bacteriophages inject their DNA directly. They don't need to uncode it. They don't need to remove the capsid. Versus animal viruses, they bring that whole capsid in, so they need that extra uncoding step.
Number 4, describe the response of the virus particles, and then no different structure types. Is it different structure types, like the envelope, the spike, the capsid?
Right, so different, diverse forms and then structure types. So just knowing that you've got the main shapes, 3 main ones that we really looked at, the icosahedral, the helical, and then the complex. And then knowing that you can have additionally, with the icosahedral or the helical, they could be enveloped or non-enveloped. When they're enveloped, it kind of masks their shape. It doesn't mean they don't have a shape, right? But the shape is referring to the capsid, not the envelope. So, so you have that additional situation, and then, composition is protein and nucleic acid at minimum, and then additionally some have lipid and the spikes, they're usually glycoprotein spikes.
I know for number 8 you said that the virus punctures the cell wall or membrane, but can the cell ingest the virus, eat it, or?
Right, so number 8, and I said don't worry too much about all the details of viral entry, and more as it kind of cross that out or add, plus no viral multiplication, all the steps. So when you know viral multiplication steps, that kind of encompasses the entry as well, which for animal viruses we looked at 2 main ways they could enter, and one of them is, the cell basically ingests the virus by an endocytosis, which endocytosis includes phagocytosis and pinocytosis. So they can be ingesting them, or in the case of the envelope ones, the envelope might fuse with the membrane, and then the cell would bring it in, or would get in through that fusion. We're not going to have time for Jeopardy. I can't add those questions, but we can look at a few Jeopardy questions, I guess, if they want us to do that. But I guess we'll just ignore that last category. But thank you, whoever wrote them. If you give them to me, I might